Okay, Biol115, welcome to the lecture on biotechnology. Skills to master for part 1, where we cover recombinant DNA, also known as rDNA. We're going to define all of the relevant terms, and a few are listed here, including cloning, gene therapy, genetically modified organisms or GMOs, transgenic organisms, sticky ends, plasmid, vector, chromosome, genome, and gene. And some of these you should already be familiar with because we've been talking about genomes and genes for quite a while. So remember, gene is a particular region of a DNA. A lot of times will encode a protein, but it could be regulatory like the promoter or the operator. And the genome is the sum of all of the genes in an organism. We will also describe features of recombinant DNA, which can be abbreviated rDNA. So the little r stands for recombinant. This is different than little r RNA. Little r RNA stands for ribosomal RNA. This is recombinant DNA. So be very careful when you're writing this out because one letter makes a big difference here. We're also going to explain and describe the process to create an rDNA molecule, including the functions of DNA ligase, which we already talked about, and restriction enzymes. Those are new. We will also be able to calculate the size of an rDNA molecule given a vector and a gene size, and you're going to love it. It's so simple. Not tricky at all. Okay. DNA cloning creates multiple copies of the same DNA, and we've already talked about one process in which this happens, which is DNA replication, one of the steps in the central dogma, where we can take a DNA molecule and then make two copies of it. But for biotechnology, we don't have to copy the entire genome. That's DNA replication. Cells do that for us. What if we just want to copy a specific region, like just one operon, or one gene out of an operon, one structural gene that encodes a protein? We can do that using DNA cloning techniques. A lot of times, the reason why we would want to look at a specific region or a specific gene is to learn more about that region or particular gene for scientific research. So this can give us insight into, for example, diseases or protein functions, that sort of thing, so we can understand our world a little better. Additionally, beyond research, occasionally people want to modify organisms in order to achieve some benefit. So an example of that would be making recombinant DNA or rDNA. So we take recombinant DNA. Well, what recombinant DNA means, let me back up here for a second, is you take DNA from more than one organism and combine that together. So for example, what if there's a gene in one organism that allows that organism to live in extremely cold temperatures? This happens in insects. And we want that gene to then be put into a tomato plant to help tomato plants survive at cold temperatures. We would make recombinant DNA and insert it into that tomato. That tomato would then become a transgenic organism. So trans means across, genic refers to genome. So these are organisms that have genes across species. Transgenic organisms. Okay. And then gene therapy, because people don't like to be considered themselves transgenic, because transgenic organisms have this very bad connotation. Many people hear the words GMOs and they feel afraid, they feel that it's very bad. But this same process, making recombinant DNA and giving it to humans, is what we call gene therapy, and this has saved hundreds of thousands of lives. So it's not all necessarily bad, but this is why we have different terms for humans, gene therapy, and for other organisms, transgenic organisms. And I would also like to add here that any modification to the genome is considered a GMO, a genetically modified organism. And as we're going to talk about later, you've actually been consuming GMOs if you've ever had any fruit or vegetables from the store. You've been consuming GMOs probably your entire life. But we'll talk about that later. So this is where we can take one copy, and through research we can make actually multiple copies, hundreds of thousands of copies, whereas in the cell you just make 2 copies at a time. Okay. So in order to make these recombinant DNA molecules, we can use bacterial chromosomes, vectors, or plasmids. So the advantage here, which we've already learned about, is that the genes in bacterial chromosomes are organized into operons, like the lac operon, like the trip operon. And this is a huge advantage, because not only do you have to insert a gene into a genome to make a transgenic organism for some benefit, but you also have to be able to turn that gene on and turn that gene off. If you cannot control when that gene is expressed, it will very likely destroy the cell. So this is where bacterial chromosomes are really, really useful, is because they have these very simple operons with simple molecular switches, so we can turn these genes on and turn these genes off again. Okay. So you remember that bacterial chromosomes are the entire genome of prokaryotes, and most of the time they look like a large circle. Bacteria have circular chromosomes that look like an O. Your chromosomes, when they're condensed, look like X's. So that's X's and O's, like tic-tac-toe. Chromosomes in bacteria sometimes also can be linear. They look like a straight line. Or they could be very, very tiny. So small regions that are detached from the main chromosome are called plasmids. And these are extremely useful because they're self-replicating. So these still will replicate themselves in a different host organism, and it can make multiple copies of your genes by themselves. That's independent from the main genome that's required for the organism to stay alive. These are like extra bonus DNA. Okay, so that's chromosomes and plasmids. So we can take these plasmids and use them as vectors. So vectors means to carry. So we can take these plasmids and use them to carry a new gene. And these are still organized into operons, and that gene can still be expressed. And the advantage here is that those are self-replicating and they contain operons. So we can turn the gene expression on and off. Very useful. Okay, so why is this possible? Why can we introduce bacterial DNA into other organisms, and vice versa? How can we take other organisms' DNA and introduce it into bacterial vectors that will carry our gene around and express that gene? So think about this. This is a really great question. So we know that each organism has its own unique DNA sequence, but consider this. Does the chemical structure of DNA differ between organisms? Chemical structure. So we know the DNA sequence is going to differ, but do the DNA bases differ? Are there special DNA bases for eukaryotes? Are there special nucleotides in eukaryotes? What about prokaryotes? Did we learn different nucleotides for prokaryotes and eukaryotes, or were they absolutely the same? So think about this and ask during lecture if you're confused. Oops. Okay. So 2 enzymes are needed to introduce foreign DNA, which would mean like a gene of some particular interest, or GOI, into vector DNA. The vector DNA will then carry this interesting gene that we're going to research for whatever reason. These 2 enzymes are restriction enzymes and DNA ligase. So we already talked about DNA ligase. DNA ligase is like the tape that fixes the covalent bonds in the DNA backbone. We saw that during DNA replication. Restriction enzymes are like scissors. So these cut the DNA. They cut the covalent backbone bonds, and then this ligase can come in and fix those breaks. Okay, so the way that this works is restriction enzymes are scissors, recognize specific DNA sequences. So they go and they search around until they find the specific location where they're going to cut the DNA backbone. So the first step is finding the DNA sequence. So they just kind of mosey around, scanning the DNA until they find the region where they bind. Then they will hydrolyze the covalent bonds in the backbone. So that means they're going to break the DNA backbone. And that's what this little dotted line means. So remember, when we depict DNA like this, the backbone is on the outside and the bases are paired on the inside. So here's our non-covalent bonds in the middle, and the covalent bonds are on the outside. Once we break the covalent bonds, those non-covalent bonds can easily break and separate these two strands of DNA. Because remember, this is like Velcro. These interactions are like Velcro. Okay, so these DNA, this creates two DNA fragments with sticky ends. And what sticky ends means is that this has unpaired DNA. DNA likes to have two strands. It is most stable with two strands. So it being one-stranded is unstable, and this would prefer to be hydrogen bonded to something. So this single-stranded DNA is going to search around until it finds something it can base pair with, and that's what makes it sticky. And once it finds something, then it's going to stick to that. So sticky ends, single-stranded DNA that occurs after restriction enzymes. Okay, this gives us a huge benefit. So here I've drawn in the backbone as these solid blue lines. This DNA ligase repairs DNA backbone breakage. Right? So our advantage here is that we've separated these two strands of DNA. We can take a new piece of DNA, shown here in purple, and as long as it is complementary in the sticky end portion, this will stick together. DNA ligase then comes in and repairs the backbone breaks. So this is exactly how we get recombinant DNA. Okay. So as long as we use the same restriction enzymes, it'll generate the same backbone breaks, the same sticky ends, and it will be complementary. Okay. And we can do this with the other side as well. So here's some other piece of DNA that we can then use DNA ligase to fix that up as well. And now we have 0 sticky ends left, everything's paired up, and we've got new DNA ligated, attached to our vector DNA. Okay, so here's another way you can think about it. So as long as our restriction enzymes are the same, we can use them to cut the gene of interest and the vector. So red here is our gene of interest, blue is our vector that we're going to carry the gene of interest. So we cut, and then these pieces of DNA will actually diffuse throughout the solution. Eventually this will end up in the correct spot, and our ligase will then fix it into place. And this is our new recombinant DNA molecule made from vector DNA and a gene of interest. So, now let's talk about how to calculate the size of recombinant DNA. So, if the vector size is 600 BP, this means base pairs. So that's like on a single strand, one base, and then on the other strand is the second base, right? So base pairs is just, you count the nucleotides in one strand of DNA. So this is how we measure DNA size. So this is 600 base pairs, and the gene of interest is 100 base pairs. What is the size of the recombinant DNA molecule made from these two? So we have our vector. We know that this is 600 base pairs that's given in the problem. We have a gene of interest. We know that this is 100 base pairs. Somehow we're going to use our restriction enzymes and ligase to cut these and then combine them. So what is the final size of the recombinant DNA? And for this, you must remember you are taking this gene of interest and adding it to the vector. So therefore, the size of the recombinant DNA will always be the size of the gene of interest plus the size of the vector. So in this example, 600 base pairs plus 100 base pairs. This would be 700 base pairs total. Nothing tricky there. Okay. In summary, recombinant DNA. We use restriction enzymes and ligase to make recombinant DNA. We start with plasmids because they are self-replicating and contain operons as a vector to carry this gene of interest. So we can take a gene from a human cell and use restriction enzymes to cleave that DNA here and there. So in this example is insulin. So this is actually how insulin is made for diabetics. So we can make a whole lot of insulin, and then they can inject it because their pancreas doesn't work so great. So we cut the gene of interest, the insulin gene, and a bacterial gene. We put these two together, the DNA ligase seals the human gene and the plasmid, making recombinant DNA. This is then put into a bacterial cell to make multiple copies, and which will give us a lot of insulin genes, which will then give us lots of insulin. Insulin is a protein. So the rDNA in the final step is put back into a host cell and replicated there through gene cloning. And that is how we get insulin. The end.